Hello, curious minds, and welcome to the very first real episode of the HEAS podcast. I am your host, Victoria Oberreiter, and I'm thrilled to take you on a journey through the incredible world of human evolution, archaeology, and all things scientific. We are bringing you a captivating lineup of brilliant speakers, each with their unique insights and experiences to share. This week, we have on Clément Zanolli, a paleoanthropologist working at the University of Bordeaux. He specializes in dental morphology of ancient hominins and talks about partaking in exciting excavations and his findings in Southeast Asia. Next up is Lucy van Dorp, who is a group leader at the University College London. Lucy is working on understanding the epidemiology of infectious diseases using both modern and ancient genomics. She has also done a lot of research on coronavirus strains. Last but not least, we have Takis Karkanas, a distinguished geoarchaeologist from Athens, Greece. Takis has done a huge body of research on archaeological site formation processes, and he specializes in archaeological sediment micromorphology. You will be stunned to hear what we can learn from looking at the sediments our ancestors left behind for us to analyze. But before we dive into our fascinating sessions, I'd like to offer a quick heads up to our listeners. This podcast has been an adventure from the very beginning, and we're still learning the ropes as we go along. In fact, we've just received our brand new equipment at the time of the arrival of our last speaker, Takis. So please bear with us as we continue to fine-tune our production process. We promise to reward you with crisp, top-notch sound quality in future episodes. As I mentioned in the pilot episode, our guests are visiting our institution at the University of Vienna and are giving a talk at the HEAS seminar series. You can go to our YouTube channel to watch all these talks. I will put all the info, including our guest social media handles, their websites, and the links to our HEAS social media pages into the show notes below. Now, without further ado, let's kick off this episode and get ready for fascinating discussions. Welcome to the HEAS podcast. Welcome, Clément, to the HEAS podcast. Thanks for sitting down with us today for our very first recording of our new podcast. Earlier today, you already gave a fascinating talk about hominid diversity in Asia during the late Pleistocene. For our listeners, that means that they can go to the HEAS YouTube channel that I will put into the show notes below and listen to Clément's talk. But now that you're already here, would you mind giving us a brief introduction to yourself and your scientific field?
Of course, yes. So I'm a paleoanthropologist. I'm studying human evolution. And more specifically, I'm interested in a number of topics. I worked from the Miocene apes to the recent evolution, but my main interest is on the emergence of Homo, of the genus Homo. And I also work a lot in both Africa and Asia to better understand the human evolution during the early to middle Pleistocene, and now a bit more on the late Pleistocene as well.
Okay, that's a quite broad time span.
Yes, it is.
Okay, so as I said, we have a bunch of follower questions, and they're a bit broader because also people who are not from the field wrote in some questions because it's a very interesting topic. Let's get right into it. The first question is, what is the finding, research result, human remains, etc., that you least expected?
Well, I think in my case, the Kalao cave material was probably the biggest surprise. And especially so because I was lucky to be there when they found the first remain, the metatarsal in 2007. And I remember very well the face of my colleagues, couldn't understand what it was. And it was only 7 years after or 5 years after that we found more remains, especially the teeth, and we could actually understand that it's a different species than modern humans. And then from there we analyzed the remains, and I was very lucky to be part of this team. I was really excited. And for me, it was the biggest surprise because we didn't expect another species than modern humans to reach the Philippines before 50,000 or so. So
that's pretty cool. So what's the date of these remains?
So there is a minimum age. The date is around 67, 70,000. But it could be a bit older, like 100,000. It's not very clear because we don't have very secure, like constrained dating. But we know that it's older than 67,000.
And what species would that be?
So we name it Homo luzonensis. It's very likely closely related to another species, which is also from another island, not from the Philippines, from Indonesia, called Homo floresiensis. And the few studies we have published now suggest that both species derive from a species that was there in Asia, in Southeast Asia, that was called Homo erectus.
Next question. Which controversies surround the timeline of Homo sapiens' arrival in Southeast Asia?
I think the main issue until now was dating and stratigraphy. The integrity of stratigraphy. So many of the findings were found, many of the fossils were found years ago when the techniques for dating were not maybe optimal, and also in some cases there was not a real excavation. The proper excavation with documentation of everything. Now we have more secured datings and also sites which have been with a clear stratigraphy from top to bottom, and in which we have several also methods applied to date the remains, like at Tampaling, for example, in Laos. I work with the team working there, and it's a fantastic site with a lot of data now after almost 20 years of excavation. So, things are progressing also in this field, and now we have less controversy regarding some of these fossils.
As we all know, especially Tom, redating is a very important thing to do,
Absolutely.
Especially when it comes to excavations that have taken place like 20 years ago, as you said. Do you sometimes wish you could go back and like redo it?
Well, yes, some of the sites, but the problem is that even if we were to I'm always thinking about Sterkfontein, which is not in Asia, in Africa, this time in South Africa. It was excavated long time ago, sometime using dynamite to blast,
Cool.
Yes. Because at first it was not a paleontological site. It was a site excavated for the materials there, the sediment that was used to extract gold. So, of course, I would love to see how it was at that time, maybe excavate it more properly. But again, even nowadays, working in these sites with some geologists, they still struggle, to understand the stratigraphy, because it's not easy to understand cave sediments and sedimentation and everything. So I think even now it will not be so easy.
I agree. As an anthropologist, I usually have problems understanding the stratigraphy, and I'm always very glad if an archaeologist is there to give me some guidance, because they see 3 layers where I see 1. So yeah. All right, so let's move a little bit into dental morphology.
Mm-hmm.
Have you had the chance to look into the dental morphology of hominin hybrids?
Well, the question is, do we know hominin hybrids? So the only one that comes to my mind for which we have DNA would be the Pestera specimens. And I didn't get the chance to study properly the teeth of these specimens. But now for other material, I think in Western Asia, probably the Kavze material, some of the specimens there could be hybrids, because they have very unusual features for modern humans, with some kind of mixed intermediate Neanderthal features. But we cannot say for sure that they are hybrids.
Right, that's where the question actually starts, right? Okay, one more dental morphology question. Are there any regionally based dental differences in comparative specimen, that specimen coexisting temporally? So for example, Asia versus Africa?
So sorry, could you repeat the question? I'm not sure to have understood what you mean there.
Yeah, I actually had to write to this person and be like,
okay,
could you elaborate on that?
Okay.
So I guess the question is, species who are coexisting temporally,
yes.
What are the difference in dental morphology?
Okay. There are differences. Today, I showed in my talk that there are probably around 6 species living at the same time in Asia. And you can find differences and you can identify the teeth of each of these specimens that were found. Some of them are difficult to identify in terms of species because we are not sure, especially for Denisovans, simply because we lack the reference material. But Homo erectus, modern humans, Homo floresiensis and Homo luzonensis, I mean, it's quite straightforward to identify them, Neanderthals, so not a big issue, and we can clearly see differences.
Mm. Also, I think it's very clear when you look at the size already, right?
Yeah.
Okay, as you said, there were at least 6 species coexisting in Southeast Asia, right?
Well, in Asia, let's say.
In Asia.
Yeah.
How many more species or groups do you think we will identify in the future?
Difficult to say. If you look even a few years back at the beginning of the 2000s, Homo luzonensis, Homo floresiensis didn't exist, Denisovans didn't exist. We had only Homo erectus, but even then it was not very sure that the latest erectus survived until the late Pleistocene. Modern humans it was very debated when they first arrived, and dates earlier than 50,000 were considered to be dubious at best. And yeah, even Neanderthals, the Neanderthals which are on the west of Asia were not very well known. So at that time you would have identified maybe 2 or 3 species at most. Now we are at 6. So who knows, maybe in a few years we have 8, 9, I don't know.
It could also very well go the other way around, right? Because sometimes, as you mentioned in your talk, species are given a new species name, and then moving on as science moves on, you see that it's actually the same. So
it's true. It's probably unlikely for Denisovans, modern humans, and Neanderthals because we have the DNA, and the DNA suggests that the differences that we observe are compatible with what we would expect for species differences. But even then, the biological concept of species is very debated. Nobody knows exactly how to separate or distinguish species in a very accurate way. And especially, it's more complicated when you work on fossils for which you don't have DNA or molecular information. So it's possible that, for example, Homo floresiensis and Homo luzonensis, or if we find more material, we realize at some point that they may belong to the same species. So it would be Homo floresiensis, which would have priority. But Homo erectus would still be Homo erectus, I think. So if it were to go down, maybe of 1 species, possibly, but probably not so much.
Yeah, I guess that's something that has happened in the past, but more often than... Okay, thank you. So you mentioned the distribution of Neanderthals towards the east of Asia. Mm-hmm. What do you think? How far can we go? What was the distribution of Neanderthals?
Well, it's not necessarily to the east of Asia. I would say they reached, places like Klagnikov, near the Denisova cave. But so far, we don't have any evidence of Neanderthals into Eastern Asia or Southeast Asia. Mm. Now we found some fossils which look like Neanderthal in some ways in Asia, in China or in Laos, the tooth that we identified a few years ago. And they could be Neanderthals, but by comparison with what we know from the Denisova cave, and for which we have DNA saying that the few permanent teeth that we have are Denisovans, actually they are more similar to the material, most of the material we find with Neanderthal traits in Asia. So it would mean that because Neanderthals and Denisovans are cousins, are genetically closely related. The Denisovans probably occupied most of Asia, while the Neanderthals were mostly in Europe. They met at some point in the middle, but it's not very clear if the Neanderthals went to the east, or if even Denisovans went to the west, towards Europe. So far, there is no evidence for that.
Right. Okay, now let's go to one broader question, but I think it's just as interesting because sometimes you have to zoom out. Mm-hmm. So besides the availability of natural resources, what other factors could have influenced or motivated the geographical spread of our ancient ancestors?
Curiosity. I mean, we are different from most of the other animals because we have a culture, and we have cultures, we have tools, we communicate, we speak, we have very different things, we have different features compared with most of the animals. So I would say that at some point where you live somewhere and you see, for example, what's behind this mountain, or we see an island there, but what's there? So you want to go, you want to explore. And I think it's only natural for a very species that, or for the human species that were very mobile. They dispersed from Africa to Asia and Europe and so on. So, there is nothing that stopped all of these human ancestors. They went to very cold climates, to the mountains, to the tropical environments. They are pretty much versatile, and they can go everywhere. So I think curiosity is probably the main factor, if I were to guess what it is.
That is such a good answer, because
I don't know if it's a good answer.
I think it's a good answer, because also if you think about what sparked science, it was also curiosity that drives science and, maintains the interest of people. So I think it's a very good question. All right. Are you ready to go in a different direction? That was just a warm up now.
Okay.
What's your take on the Asian origin theory?
I mean, of modern humans.
Of all humans?
Yes. Well, I can speak about teeth, and from what I know about human evolution based on teeth, I don't see any modern features in the very early, like Middle Pleistocene material. There is no modern-like features, but I do see some of these features in Africa. So if we as modern humans, as Homo sapiens, emerged somewhere, we should find in the fossils, which are probably around the Middle Pleistocene, some features which should resemble at least a minimum what we see in the nowadays populations. And I cannot see that in the Asian record, but I can see it in the African record. So I think most of this theory emerged in Asia because they were finding a lot of fossils. They didn't know what to do with them, like what we call archaic Homo sapiens or archaic hominins from the Middle Pleistocene. And actually now we are, if you followed my talk, you will probably guess that my view is that most of these fossils could be called Denisovans. Not all of them, but many of them. And some of them are probably late Erectus. And they are, again, different from dental morphology compared with modern humans. So I would not see a direct evolution from some hominins in Asia to modern human populations.
Okay, so it's a lack of morphological evidence.
I would say so. And I don't know, but I'm not a geneticist, but what I know from the genetics is that all the evidence also is saying that, the origin is in Africa. So.
Yeah, that's sometimes not enough. So,
no, but if you combine morphology and molecular data together, then...
Exactly. Not for me or for us as researchers, but there are always voices who are speaking.
Yeah, but then it's not science. I mean, you have belief, you have other things that to take into account. But if you only consider science and the facts,
Exactly.
then we come from Africa.
Great statement. Okay, let's move on to another segment, which is called the HEAS Quick Takes.
Okay.
So it's a segment where I will ask you rapid fire questions and you answer with the first thought that comes to mind, okay?
Okay, I will try.
Let's do that. What got you into science?
Fossils. I was always fascinated when I found my first fossils in the limestones in Normandy. It wasn't, of course, even not vertebrates. It were some mollusks and weird fossils, but from millions of years ago, 70 million, 100 million. But I was fascinated by that. I always wanted to know what they were doing there, what was the life at that time. So, yeah.
It's definitely fascinating. And what still excites you about science? Every day?
I think because I always find things I don't expect. And I like this aspect, finding new things, and most of the time things I don't expect. Yeah.
Are you a cat or a dog person?
I have a dog right now, but yeah.
What's his name?
Saji.
Nice. Okay. What did you want to be when you grew up? I think you answered that question with a...
Paleontologist, yes.
Amazing. So that's a dream came true.
Yeah.
What's one of the best pieces of advice you've ever received?
That's a tough one. Several ones, but I would say the one I will always remember is that there is absolutely no limit. You can do whatever you want as long as you want to do it.
Who would you like to have dinner with, dead or alive?
Well, I mean, I've been working for my PhD on collections at Senckenberg Museum, and I was always fascinated by the paleontologist who assembled this collection. His name is Ralph von Koenigswald. And everything he wrote and published, I actually found out that it's very accurate and mostly true. But he did that from the 1930s to 1980s. And he didn't have access to all the instruments, the modern instruments that we have. He didn't have access to DNA, anything. And just based on morphology, he was able to mostly understand everything that was happening. It was fascinating to me.
That's impressive. So you would sit down with him?
Yes, I would love to
cheers.
chat with him. Yes, absolutely.
Well done. What's a French word you think is really hard to pronounce?
I have no idea. I'm French. So
You're French.
For me, it's not difficult to pronounce. I have no idea, actually. I never thought about it. If you ask me for a new word, I don't know in which language, but probably I could find one, but in French, it's more difficult.
It's probably true. I should paraphrase this question. Do you know any German word?
Well, I'm not so much versed into German, so
don't worry.
Actually, no, I don't know.
Don't worry. So linguistics.
Oh yes,
is not your.
Yes, no, yeah, I know. Entschuldigung. I'm never sure how to pronounce it because.
Perfect.
I'm starting to learn the basics of German, but yes.
Well done.
Okay.
If you were to write a book, what would it be about?
I guess about human evolution, but I'm not ready yet to write a book.
Maybe you should consider it at some later point in your
It takes
career.
too much time and energy for me. For now, I don't have the time. But.
Okay, last one. If you got a plane ticket from me right now, where would it go to?
For fun or for science?
You decide.
Well, for fun, definitely to Japan. I love the Japanese culture and the country. And for science, well, there are different options, but between Africa and Asia, either South Africa or some Southeast Asian country.
All right. We have another very quick segment. It could actually also be just a quick question, but if you want to elaborate on that, I'm very happy.
Okay.
It's called Guest's Guilty Pleasures.
Mm-hmm.
So, what is your unexpected interest or guilty pleasure that may not even be related to your scientific field? If you have one.
Well, it's related to my scientific field. As I said, I love fossils, so I love to go fossil hunting, or at least go to the field somewhere, even in France, or and look for fossils. Yeah.
Okay, and last question. You did very well so far.
Thank you.
Where can people find you online? Would you be willing to provide your social media handle
Sure.
for work?
Yes. I'm not so active, but I have a Twitter account. So sometimes I tweet my papers, my most recent papers. But I have to say I'm not so much of a social media person, so I'm not so active on social media.
Clément, thank you so much for sitting down with us for the HEAS podcast.
And thank you. Thank you for the invitation.
Welcome to our second HEAS podcast, this time from a speaker from our genomics seminar series, Lucy van Dorp from UCL.
Thank you for having
me.
UK,
who specialises in microbial genomics and population genetics. And the first part of the interview, we will be asking you broad questions from posts on Instagram. So with the general public asking you questions, and we picked a couple which we thought might be interesting to ask you. And the first one actually is, do you think that ancient pathogens that are re-emerging from permafrost as the climate changes could be a potential threat to human health?
I think very difficult to say. I think it is probably true that things are emerging from permafrost. We know that to be true. To the extent to which they can be a threat is very, very difficult to ascertain. So there may be cases where we have microorganisms emerging, which are perhaps genetically similar to something today, which causes a problem, and perhaps those would be ones that we'd be concerned about. But perhaps we have completely uncharacterized or unknown species that are also emerging, and then it'd be very difficult to sort of suggest, based on just their genomic data, whether they'd be a problem for us in the future or not.
Yeah. I think a lot of people understand this topic a bit like, it's a fantasy, it can't happen. But we've seen it happen in literature. Viruses, reactivate. Or, there's also a definition of what is a virus to begin with. Is it living, dead? What does it need to be active? And so, obviously, do you think there's merit in putting more research funding and more research in actually studying what we're getting out of compost before it gets into the oceans, before it gets into maybe the wider?
Yeah, I think we do have the toolkit to do that. So the question then is about resource and funding, whether it's the right thing to do. And I think we are already facing many potential infectious disease threats right now before we have any changes. So it's an interesting question to what extent we should be trying to future-proof versus trying to tackle what we have now. So I think if there were some good examples where we really did find that there was some emerging bugs that we would be concerned about in terms of their phenotypic properties, how they might cause us infection, or how they might cause infections in significant, say, crops or animals or so on and so forth, then perhaps that would be a really good use of resource. But I think actually our understanding of what is currently here in this current climate is also fairly poor. So I think it's a challenging question to answer, actually.
Yes. As you pointed out in your talk, we barely have any genomic data in what we have already.
Yes.
So the second question, we're going to go straight to now. How quickly, and I think it's very topically to what we just talked about, how quickly can microbial resistance spread, and can you give an example?
So antimicrobial resistance, so this is the ability of a microorganism to evade treatment. So that could be by antibiotics, for example. That's the kind of most common example. Is an emerging problem. It is a growing problem. It's going to be a problem going forward, and it can spread really, really rapidly. So one of the characteristics that is often a very common feature of antimicrobial resistance is that the genes carrying that resistance are on mobile genetic elements. So these are elements which are not particularly fussy. They can often move between bacterial species using various vehicles, which might be, for example, transposons or plasmids. And so this is something which can emerge fast. So not that long ago, we worked on an outbreak of a type of bacteria called Klebsiella pneumoniae. It exhibited carbapenem resistance, which is a resistance to one of the last line antibiotics that's used in hospitals. And we were essentially tracking an outbreak that involved this particular resistant strain of Klebsiella. And we found by tracking just this resistance gene element that it was moving really, really very quickly around the hospital and between different strains over the course of the 13 months we looked at. And one thing that was quite important in that case, and this is about thinking how these things are moving and spreading and perhaps causing more and more problems as time goes on, is that we found many cases where, for example, the bacteria was present in, say, healthy carriers or on equipment or being carried by staff. And so it's not always just the case that nasty drug-resistant bacteria exist in hospital infections. Actually, we can be harboring and carrying them and passing them on quite easily all the time. So it can be very, very fast.
So I guess co-infections would be a big topic there as well when we talk about mobile elements. And
yeah, the ability to exchange. And the reality is that some bacteria tend to share more than others, and different bacterial genomes behave in very different ways. But certainly drug resistance is one trait that is really important to track dynamically across kind of the wider environment. So that might be in healthcare settings, it might be in the community, it might be in livestock, it might just simply be in wastewater or sludge or sewage.
You've talked a lot about how you work in both ancient and modern DNA, and how the interface between those two parts of the DNA spectrum, which in the end is kind of the same thing, but just from different sources, how that works. And I was wondering, from your perspective, what are the biggest gains modern DNA is getting from ancient DNA, and maybe in what way modern research isn't making enough use of what we're producing in ancient DNA? What new ways could there be where we could take advantage of what's being generated now in large amounts and lots of funding as well from ancient DNA studies?
Yeah, I think that's true, and I would say my work kind of sits somewhere in the middle. So a lot of my work is on kind of contemporary infections, contemporary outbreaks, kind of real-time response, but also this very looking back perspective. How can we understand the evolution of the things that we're currently carrying, currently facing? And I think there is a lot of room for dialogue there that could inform each other. And some particularly good examples, I guess, is where often when we look back at pathogen genomes, we may well find lineages which are, for example, today extinct. Now, these may have been really important lineages in terms of understanding the evolution of the bacteria, virus, or pathogen. So this is something that we've seen multiple times. So for example, I was involved in some work looking at plasmodiums. This is the agent that causes malaria, Plasmodium vivax genomes from medical microscopy slides in Europe. So this is malaria was eradicated from Europe in the 1940s, but these, 1960s. But these slides give us the ability to look back, and in doing so, we were able to rediscover that eradicated European lineage and understand that was likely a vital kind of time point for understanding the spread of malaria, for example, to the Americas. In my talk today, we talked a little bit about some lineages that we've seen in the herpes simplex virus 1 as being a really important hallmark of where we might have lost lineages in the past, and maybe what we experience today isn't a good representation of everything that was circulating. So I think that's really important in understanding the wider context, understanding what are the drivers of the evolution of pathogens. Another really important aspect of looking back is that by looking back, we get direct observations of genomes at points in time. And that can be really, really useful for estimating, for example, mutation rates, so how fast a particular organism, a bacteria, a virus, for instance, is evolving, simply by saying, okay, here we have some observations at this time, then we have some at this time, then we have some at this time, and we can simply extrapolate those to estimate how fast something is evolving. And of course, how fast something is evolving is really, really important. It can tell us, for example, how concerned we should be about the emergence of new variants or lineages. It can also tell us how quickly, for example, vaccine targets might be evaded. And it might also tell us some really important properties about regions of the genome that we know are implicated in working to or against the host immune system. Have those been the same over time, or have they changed, and how quickly are those things changing?
I mean, the disease landscape, the human one, is always evolving. We've seen it very clearly the last couple of years with the emergence of the new pathogens, which does happen much more regularly than maybe general everywhere. There's always this question about, are we expecting it to change much more in the future, and if what will be the main driver of that?
Yeah, I mean, I think it's very difficult to say if we're seeing more now because we're looking. We've got the toolkit to look much harder. I think the reality is that humans have and animals and everything else has faced a continual onslaught of pathogens, or certainly of microbes, throughout history. And so we now have the opportunity with toolkits like genomics or epidemiology or various testing techniques to track that. So I think we're all much more acutely aware. And the reality is there's probably many epidemics and pandemics of the past that we just simply have no knowledge of. That's not to say that we shouldn't be thinking about it as a growing threat. We have this toolkit, so we should be using it. And I think that raises some interesting questions. So what are the correlates, at least? It's potentially very hard to talk about the determinants, but the correlates that might be important in terms of having more emerging diseases. And There's plenty of suggestions. So some are, for example, increased urbanization, increasing contact between humans and different wildlife species. Some of that might be linked to the process of climate change, just simply changing the interactions between different animals, different hosts, providing more opportunities for microbes to come into contact with different hosts, which they simply wouldn't have before. We also should be aware that the way in which we process or farm, for instance, livestock is sometimes very conducive to the spread of infectious disease, so potentially opening up some vulnerabilities. And so I think we have to be acting like this is an increasing threat because we have the power to act on it. And I think that could involve, for example, targeted genomic surveillance, maybe looking at screening of, for instance, wastewater to try and identify new and emerging threats, but could also include, for instance, identifying particular strains that we might be concerned about, that we might be tracking their evolution, and preemptively perhaps designing monoclonal antibodies or vaccine targets with, we have very rapid vaccine technology now to think, so let's, hope we never need to use this, but if we do, we have essentially a bank of a resource to be able to tackle new threats quickly. And I think the reality is we'll never be able to do that perfectly, of course, but I think that, we do have some tools in our arsenal to be able to be more proactive in the way in which we look for and tackle new and emerging disease threats.
All right, Lucy, are you ready to move on to the HEAS quick takes, which is rapid fire questions?
I will do my best.
Let's get started. What's your biggest motivation?
Probably curiosity. Do I need to expand?
You can.
Okay. Yeah, I think, it's probably an inherent nature of wanting to do science, but just wanting to understand, know a bit more, being driven by that.
Were you good at school?
Yeah, I think I was a bit of a geek at school. I did like my study, to be honest with you. But yeah.
What did you want to be when you grew up?
I actually always wanted to be a vet, so loved animals, and I find it quite funny now that I actually work with quite a few people connected to the veterinary field in the kind of work that I'm doing on zoonotic pathogens. So in some sense, I've kind of ended up doing a little bit, but in a different way.
If you had to switch professions, would you then become a vet now?
Oh, I don't think so.
No?
I don't particularly like blood. I think it was a terrible career choice as a child, to be honest. I'm not quite sure what I would do. Probably something radically different, something artistic or creative. I'm not sure.
And what got you excited in science?
It sounds a bit cliche, but discovery, and that I think does make sound like scientists are discovering new things all the time. That's certainly not true. Discovery for me could be like just fixing that little niggly thing that I've been trying to work on for ages, and I think that is quite a motivating driver to sort of feel like a gain is made over challenges on a kind of regular basis.
Which book are you currently reading?
I'm reading a PhD thesis I have to examine, and there's little time for other literature, I'm afraid.
I can imagine. I was going to ask you, and can you recommend it? But I guess
I will let you know.
Good luck for the PhD student. Who would you like to have dinner with, dead or alive?
It would actually bizarrely be my grandfather on my dad's side, because he was also a scientist, and I never got to meet him. He died before I was born, so I'm always a bit interested because around my family network there isn't generally an interest in science aside from that individual. So it would have been nice to have dinner with him.
And do you have any science-themed home decor? Maybe something that you inherited from that side of the family?
Nothing from that side of the family. I do have on my desk at work, so it's not at home, I do have an old microscope, which was a teaching tool used at UCL that was going to get thrown out, and it's ended up on my desk because I have a bit of a pet interest in trying to obtain genomes from microscope slides from the past. So it's a non-functioning old but quite beautiful microscope.
That's really cool. When we moved to this building, I took the chance and I got my hands on some nice old stuff from the department as well, so I can relate. And that's actually in my home.
Okay. You got it home. This one's too heavy.
All right, so let's move on to our next segment. It's called Guest Guilty Pleasures. So do you have any unexpected interests or guilty pleasures that might not even be related to your field?
I'm quite into baking, I don't know if that's unrelated. I think actually quite a lot of scientists probably like baking because it's quite methodical, but I do like to bake.
What do you like baking? What do you make?
Mainly cakes. I'm all about the decoration, I have to say. So sometimes they don't taste great, but they look really nice.
They look. One of our follower questions was about ancient pathogens. And as the climate changes, that they might be or are actually re-emerging from the permafrost. And if you think about it in that regard, they're basically coming back to life. I'm doing quote unquote. Do you consider a virus a form of life?
Well, this is a big question. It does kind of come down to what you define as life. And for a virus, it's a replicating entity that needs a host. I guess there's plenty of us that might say we persist and exist because of the support we have from systems around us, and maybe to a virus that is its life too. And so, in that sense, maybe we could consider a virus as alive, but I think it's a very challenging question to answer, and probably doesn't have a good answer. Actually, it's more of a philosophical question than a scientific one. It depends how you define life.
Exactly. Let's do one more segment. Travel through time.
Okay.
So that's a segment where we ask our guests to pick a specific historical era or a site they'd like to visit, or if you would have a time machine, let's put it that way. To which era would you go? What would you like to see?
I think I would like to have existed or go back and see a period where Homo sapiens weren't the only genus Homo on the planet. And it would be very fun, it's endless speculation on what Neanderthals and Denisovans looked like, how they interacted, friend or foe. And I would love to just jump on a time ship and observe and get a sense of what that dynamic was like. And yes, but I guess part of the reason that that's so fascinating is because that idea has spawned endless stories and thought. And perhaps it's nice to keep that as a speculation as well.
Very good answer. Okay, now to wrap this up, where can people find you online?
So I have a X or Twitter account and a website, and my email address is on the website, so I'm happy to chat to people or take questions there too.
Thank you so much for sitting down with us for the HEAS podcast.
My pleasure. Thanks for having me.
Welcome, Takis, to the HEAS podcast. Can you give us a little introduction to yourself in your scientific field?
Yeah, I have been trained as a geologist, but early in my career, I moved to archaeology, and I'm applying the methods that I have been trained in geology, and the field that I'm now specialized is called geoarchaeology, and I'm using different methods. Of course, my favorite method is micromorphology, soil and sediment micromorphology.
And why did you choose microbiology as your main line of research? I think during your talk that you gave earlier, you mentioned that you're an enthusiast for barbecues and you're working a lot with very ancient fireplaces now. So
yeah, okay.
I guess it was also a joke, but it was very fun.
I didn't start using micromorphology because of my barbecue involvement, but I used micromorphology because early when I was studying, I was involved in some archaeological studies. I thought, okay, what I can offer with my experience in geology, and I saw that actually people were using already thin sections to study sediment, and because I believe that it's crucial to know what you are analyzing, the thin section, the micromorphology gives you this opportunity to know what exactly you see and what exactly you analyze. It's actually that and of course, I knew petrography. I have an experience in studying thin sections, in mineralogy, so it was easier for me to use this type of analysis. And I have great results. So it was confirmed, let's say, that micromorphology can, the study of thin section can be used in archaeology with very successful results.
From your talk today, we also learned that many inferences can be made from looking at the micromorphology of a site, such as if the ceiling of a cave has collapsed, or if a sediment was trampled, how many times a fireplace was lit. So can you give us an overview of what we can learn from micromorphology, especially regarding human activities at a site?
Because all artifacts are found inside a sedimentary matrix, the way that the sedimentary matrix is formed tell us about the activities of the people. So in theory, since we are digging the sediments in order to find archaeological remains, all activities of people are recorded in the sediment, and we can study, and if we are lucky and there are still the evidence there, we can reveal them. I mean, we can definitely find how many times a fire has been lit. We can find evidence of damping. We have evidence of brushing, of producing structured surfaces, of maintaining of the structured surfaces, of mud floors, or making pits and dumping material, what exactly the dump material is, if they have dump material, if the material has been disturbed by later activities, if the sediment had been reworked by natural processes, by wind, by rainwash, all kind of details.
What I found was really interesting, and what I never really thought about in detail, is that you said if you light a fire once, the ashes are easily carried away by wind or other processes because it's kind of a fluffy material, right?
Yeah.
So you said for a layer of 30 centimeters of ash, about 1,000 times fire has to be lit about 1,000 times.
Yeah, that's true.
That is so interesting.
Yeah, that's actually how I started doing all these experiments, because I was using my barbecue for several years, because I like to make fires every weekend, barbecue every weekend. So I saw that even, let's say, after 10 years of using the barbecue constantly, there was not so much ash produced as someone will think based on the thickness of the ashes that are produced just by a single fire. Let's say the idea was curiosity. I wanted to explain why a thickness of a few tens of centimeters actually is the result of probably thousands of fires or hundreds of fires.
How many times a fire has to be lit to make it visible in the profile so that it stays preserved and that you then can go back and say, like, okay, that definitely was a fireplace that humans lit?
Yeah, I have seen actually that you need at least 5 or something like 5 to 6, 7 relighting events in order, let's say, the ashes to start becoming cindered and crusted. Of course, normally, okay, this is the least amount and least, times, but I believe, let's say, something like 10 to 15 fires probably could be recorded in the archaeological record. I mean, in theory, you can probably, even a single fire event can survive if it is immediately buried, but you will see just, you will produce a millimeter probably, or a few millimeters of ash remains that will be very difficult, let's say, to be excavated, observed during excavation. But yeah.
Now, a little more of an general question.
Mhm.
What were the major developments in discovery in micromorphology in the last decade?
We have made progress in various fields. I believe that uh, uh, there have been a lot of experiments with fires, for example. We have this beautiful work of study of mating in a Paleolithic sites in South Africa, Djibouti, for example, that people were making matting and they would burn them constantly. This was a nice example of having seen new, let's say, practice and activities through micromorphology. More and more, we understand small scale natural processes. We have also, and I have been involved in this, recorded micromorphologically movements in the side of the sediment of clast, making rotations that suggest deformation features related to gravity flow processes, as we call them, or material that moves downwards by gravity. I have the feeling that has been made a huge progress, to be honest, and also have done a lot of ethnogeoarchaeological research. So this helped us a lot to understand all these kind of processes, because you see what's happening in a real environment. Yeah.
So that tells you a lot about the site formation
Yeah, exactly,
processes, right? And can that also give you hints about the paleoclimate then? I guess that's probably related to it, right? Then you can say,
Yeah,
I guess you're the
okay.
expert.
Studying the paleoclimate is not straightforward because sometimes you have the same product by different environmental factors. And that's actually a lesson that I took once one on my of my first studies. But definitely, let's say you can use a micromorphology to study all these phenomena related to climatic changes. But in particular, in this case, you do need, let's say, proxies from other analysis to secure your result. You cannot say, let's say, okay, because I have, let's say, more, I don't know, rainwash, that this means that actually the climate became wetter. It can be just we have more storms, for example, which is actually it means that the climate was getting more drier. So you need, let's say, of course, it helps because you see facts, let's say, this change in the environmental conditions, but you have to nail it down with other proxies as well.
I guess that applies to science in general, that the more methods you incorporate,
Of course.
the more detailed your picture about what could have happened gets.
Yeah.
So it's definitely a good addition and can help in many, many fields. Also in ancient DNA. I work in ancient sediment DNA,
okay.
and that's what I have to ask you. So recently we've seen this Masalani paper, and it gave us a good idea, I think, of where in the sediment the DNA can be preserved.
Mm-hmm.
So what are your thoughts on where in the sediment the DNA can preserve? What do you think?
I assume, let's say, the first thing that I will look is probably the clay fraction. If actually the molecules of DNA are sealed somehow inside the clay fraction, but I cannot, let's say, dismiss the possibility of having very, very fine fraction of bone or other material that you are interested in its DNA content contribute. So I don't know, every case, maybe both, let's say.
For the people who are not so familiar with micromorphological processes, I will explain this quickly a bit. So what you do when you want to look at a thin section is you dig around a rectangle in your profile, right? So that it creates a block of sediment. Then you plaster around it and cut it loose from the profile. So then that enables you to transport this block to the lab.
Mm-hmm.
And then you resin impregnate it, right?
Mm-hmm.
So that means from this resin impregnated block, then you can cut very, very thin sections that then can be looked at under the microscope.
Yeah.
Now to actually one of our follower questions from Instagram. Do you have any thoughts why these impregnated sediment blocks seem to preserve ancient DNA better than the average loose sediment sample?
I don't know if this is the case, meaning that it's preserved better.
I guess it's what you target in there, right?
Yeah. I believe, okay, first of all, it's strange. In a way, I don't think that have been resolved. Because in theory, let's say the resin, the temperature of resin, when it's cured, it can go up to 60, 70 degrees. I don't know, I'm not familiar, of course. We know better if actually ancient DNA is affected by such temperature. In theory, probably not. But sometimes, depends on the resin and the procedure, can reach higher temperatures. So if, let's say, the resin, the temperature of the curation does not produce any problem in the DNA, so in theory, the solidification of the material, let's say, and its preservation through time, because, the resin actually seals the whole thing and does not allow the DNA further to decay or whatever is enclosed inside the resinated block. So I assume that's why probably we have good results. And there's another thing, because normally micromorphologists target areas that and take the samples from areas that they know that have they are rich in information. So it's also probably the sampling procedure that leads to better results, rather than someone going without understanding the sediment and just take samples for DNA analysis. Because, in theory, geoarchaeology know better the stratigraphy, or can read the stratigraphy in a better way than anyone else, because he's trained or she's trained on this.
Also, when you're taking a whole sediment block, it's easier because the sediment is kept in situ. So you can still tell the layers from this intact block, and you don't just have a scoop where somebody went into the profile, like me.
Yeah. And you can target, and you see, and you can think, and you can target. That's true.
And of course, I do that with the help of the archaeologists, because as you said, and I've mentioned this in another podcast, I'm always quite grateful for archaeologists to tell me exactly where one layer starts, one ends, and then you can, of course, target it. But in the end, what you take when you just take a spoonful out of the profile is just one point, in this whole thing. You cannot go horizontally. And if you have this block with you and you target one point there, you drill one, whatever you target. Mm-hmm. And then still you can go back. You can go back and resample more because you took that whole block to the lab with you. So I think micromorphology is definitely the way to go, as we've also seen from the research that has been done.
I'm happy to hear this. Yeah. No, I understand what you're saying. And also, because I often involved in support, let's say, sampling of other fields, for example, phytolith, or similar, let's say, type of analysis. And as you said, you put the scoop and try to sample the particular layer, but most of the time you fail because their layers are thin, or as you move inside a few, just 1 centimeter, the layer changes, the sediment changes. So actually you don't, that's the problem. Still, even if you be very careful, you actually are not sure what you are sampling. Where when you have a slab of micromorphological slab, as you said, you can target exactly where you want to sample, and you can sample laterally and repeat, let's say, or not the results, etc.
That is so true. You have to be very careful when sampling at an excavation site because, it's kind of messy as well. Sediment can crumble from above. The layers can mix. And as you said, when you go in, first we have to take off the surface anyways to avoid contamination. And then the deeper you get inside, the less you can actually tell whether you're still in the same layer or not.
Absolutely, yes.
So I guess a lot speaks for sampling these resin impregnated blocks. All right, now let's move on to our HEAS quick takes. This is the part where I'm going to ask you rapid fire questions and you answer with the first thing that comes to mind.
Okay.
Don't worry too much about it. What did you want to become when you grew up?
Architect.
What got you excited in science?
I like, let's say, to find the way things work, probably.
That's the answer I mostly get, I think. It's curiosity and to find out how something works and why.
Yeah.
Are you an early bird or a night owl?
Night owl.
Do you also like to work at night?
Yeah.
What is your proudest moment? Or was? Private or work-related?
Proudest moment, okay. I believe that actually my work on this later work on fire experiment, I was very proud of it.
Now a question from me, because I need to know. What's your pro tip for avoiding back pain when excavating?
Good thing is that I don't have any problems with my skeleton, so I really make very strange position. I don't feel the pain, so I'm not the best one to ask. I'm sorry for that. I'm lucky in this way.
You definitely are.
Yeah.
I will have to keep on asking this question because for me it's not working that well. What's your ideal day off work?
Normally, I'm reading books, or I'm playing with my garden or my barbecue.
Right. You're probably going to light a fire, as you already mentioned. And what are you currently reading?
I'm reading, try to remember now. Okay, this is strange. I'm reading a book of John Locke, the philosopher.
That's not strange.
I like to read, how, let's say, to understand how we think, how you produce theories, because science is theory-laden as well. So I like to read epistemology, sometimes philosophy as well.
Now let's move on to our next segment called Travel Through Time.
Mm-hmm.
This is our segment where, imagine, I will give you a time machine. To which historical era or archaeological site would you like to go? What would you like to visit, see, and why?
Interesting question.
You cannot go to the future because we're in human evolution here, and that's the limit.
Yeah, I would like actually to go back in time and see and be part of a Neanderthal group, because I'm very, very curious to see if these people, let's say, how they behave, if they were similar to us, or, if they really, let's say, had the same way of, like the way we think, the modern humans, if they actually, or let's say, not much different from us. I will definitely want to go back then.
Now let's move on to guests' guilty pleasures. Do you have any unexpected interests or guilty pleasures that might not be related to your field?
I guess you don't mean reading or going trimming your garden, but
you can
I often do it.
It doesn't have to be very unexpected. I just like to ask people that because sometimes really interesting things and quirks are coming.
I have a garden. I often, do gardening. I forgot. I love to cook, and also sometimes experiment with cooking. And cooking is one of my favorite things, actually.
That's nice. The last segment that we're going to do is Speakers Ask Speakers, and that is where every speaker provides a question for the upcoming speaker. It doesn't have to be anything scientific. It could be whatever. They leave a question, and I'm going to ask you the question now. What do you think are the major outstanding questions in human evolution, and how will they be answered?
Interesting. For me, the major question is it's why I wanted to go back and see Neanderthals, how they behave, is to understand when whatever is called modern human behavior started. For example, language or modern behavior. But I think that the most crucial question is when will this happen and how, actually.
Thank you, Takis, so much for sitting down with us for the HEAS podcast.
My pleasure. Thank you very much.